Welcome to my American History Notes, where I share with you some things I've learned about American history and draw some conclusions you might not agree with. You might even be offended by, but my goal is that you'll have your own conclusions about American history. You'll study it yourself. Today we're talking about in the context of 19th and 20th century social movements. We'll talk about cloning. So let's talk about cloning as the creation of. And you don't hear much about this today. It was a big hot topic in the 90s. So, just going to discuss a little bit about it today. So, it's the creation of an individual that's a genetic replica of another individual. The process transfers a nucleus from a somatic non reproductive cell into an enucleated fertilized egg, one that has had its own nucleus destroyed or removed. The genes in the transferred nucleus then direct the development of a complete organism from the altered fertilized egg. Two individuals who are clones have identical genes in their cell nuclei, but differ in characteristics that are acquired in other ways. So cloning is a natural phenomenon in species as diverse as armadillos, poplar trees, aphids, and bacteria. Identical twins are clones. Biologists have been cloning some organisms, such as carrots, for decades. Attempting to clone animals has been far less successful. They began long before the February 1997 announcement of the birth of Dolly, a sheep clone from a mammary gland cell nucleus of a 6-year-old sheep. Oxford University developmental biologist John Gurdon cloned frogs in the 1960s, but in a limited way. He showed that a nucleus from a tadpole's intestinal lining cell could be transferred to an enucleated fertilized egg and support development to adulthood, and that a nucleus from an adult cell could support development as far as the tadpole stage. However, he was unable to coax a nucleus from an adult amphibian cell to support development all the way to adulthood. In the 1980s, several companies tried to commercialize cloning of livestock from nuclei taken from the embryos or fetuses. The efforts failed because the cloned animals were nearly always very unhealthy newborns and did not survive for long. Currently, livestock cloning is limited to research, although some companies offer tissue preservation services in anticipation of future advances in commercial livestock cloning. There is no reason to believe that human clones would fare any better in terms of health or survivability than most cloned animals do. Now, there was a cloning ban. The ethical concerns about whether an action is, quote, right or wrong are often clouded by subjectivity, emotion, and perspective. Cloning members of an endangered species, for example, is generally regarded as a positive application of the technology, whereas attempting to clone an extinct woolly mammoth from preserved tissue elicits more negative responses, including that this interferes with nature. A project at Texas A&M University, funded by a dog lover wishing to clone a beloved deceased pet, announced the first successful cloning of a domestic animal, a cat, in February 2002. Cloning pets when strays crowd shelters might be seen as unethical. A different set of ethical issues emerges when considering the cloning of humans, which a few scientists and physicians have proposed doing outside of the United States. Bioethics is concerned with the rights of individuals, such as the right to privacy and the right to make informed medical decisions. It is difficult to see how these issues would apply to cloning unless someone was forced or paid to provide material for the procedure, or if an individual was cloned and not informed of his origin. Ethical objections to cloning seem to focus more on the fact that this is not a normal way to have a baby. Accordingly, the U.S. House of Representatives voted overwhelmingly on July 31st back in 2001 to pass legislation that would outlaw human cloning for any reason. However, the broadness of this action may impede other types of medical research, thus introducing a different bioethical dilemma. The legislation sought to ban all human cloning, both reproductive cloning that will be used to create a baby and therapeutic cloning. In therapeutic cloning, a nucleus from a somatic cell is transferred to an enucleated donor egg, and an embryo is allowed to develop for a few days. Then cells from a part of the embryo called the inner cell mass are used to establish cultures of embryonic stem cells that are generally identical, genetically identical, to the individual who donated the somatic cell nucleus. If this person has a spinal cord injury or a neurodegenerative disease, the embryonic stem cells might specialize into needed neural tissue. To treat muscular dystrophy, the cells might be coaxed to differentiate into muscle cell precursors. Such tailored embryonic stem cells would have had many applications. The person's immune system would not reject what is essentially its own tissue. Some people argue that therapeutic cloning violates the rights of early stage embryos. Others argue that banning the research violates the rights of people who might benefit from embryonic stem cell therapy. So, according to the bill they were trying to pass back in 2001, um, it was a banned selling or producing a ban on producing or selling any embryo produced by human cloning. Scientists caught in the act were supposedly be able to be fined up to $1 million or 10 years in prison. Um, so, and before 2001, though, bans on using embryonic stem cells applied only to federally funded research. So let's um, let's just skip and look down a little bit into some cloning misconceptions. The premise that a clone is an exact duplicate of another individual is flawed. And so, if the intent of the cloning is to create such a copy, it simply will not work. For example, the tips of chromosomes, called telomeres, shorten with each cell division. A clone's telomeres are as short as those from the donor nucleus, which means they are older even at the start of the clone's existence. DNA in the donor nucleus has also had time to mutate, that is to say, it's had time to undergo modification from its original sequence, thus distinguishing it genetically from other cells of the donor. A mutation that would have a negligible or delayed effect in one cell of a many cell organism, such as a cancer causing mutation, might be devastating if an entire organism develops under the direction of that nucleus. Finally, the clone's mitochondria, the cell organelles that house the reactions of metabolism and contain some genes, are those of the recipient cell, not the donor, because they reside in the cytoplasm of the egg. Mitochondrial genes, therefore, are different in the clone than they are in the nucleus donor. The consequences of nuclear and mitochondrial genes from different individuals present in the same cell are not known, but there may be incompatibles. So, perhaps the most compelling reason why a clone is not really a duplicate is that the environment affects gene expression. Clone calves have different color patterns because when the animals were embryos, the cells were destined to produce pigment moved in different ways in each calf. For humans, consider identical twins. Nutrition, stress, exposure to infectious diseases, and other environmental factors greatly influence our characteristic. For these reasons, cloning a deceased child, the application that most would-be cloners give for pursuing the technology, would likely lead to disappointment. So bioethical concerns over cloning might be moot because the procedure was always been extremely difficult to do. Dolly, back in the 90s, was one of 277 attempts. Cumelina, the first cloned mouse, was among 15 live-born mice from 942 tries. Cloning so often failed, researchers thought, because it was not a natural way to start the development of an animal. That is, the DNA in a somatic cell nucleus was not the same as the DNA in a fertilized ovum. The donor DNA in cloning did not pass through an organism's germline, the normal developmental route to sperm or egg, where gene activities are regulated as new organism develops. Ethical objections to human cloning are more philosophical than they are practical. The very idea of cloning assumes that our individuality can be understood so well that we can duplicate it. If human cloning ever does become a reality, that this is not true would become evident. After all, we are more than a collection of genes. Okay, so cloning, that was a little bit of a weird thing to go into. We don't hear much about it anymore, and like I said, and it's a lot of technical stuff that I pulled from answers.com and other places. So we're going to start next, though, on the civil rights movement. That's our next discussion about American history, and I hope that you'll join me for it, and that we'll learn something, both of us, and maybe you'll argue with me or come to a different conclusion, but I'm hoping that you'll join me, okay? Take it easy. Let me have this. Okay, there you go.